Hello dear learners, in this video we have to talk about the gene mutations. In the previous two videos related to mutation, we were discussing about that how the chromosomal structures can have the mutation and how the chromosomal numbers can have the mutation. But now we will be interfering in the sequences of the DNA or the sequences of the gene, that there can be certain mutations which can occur in the sequences of the DNA or the sequences of these genes. Now these particular mutations are referred to as gene mutations. Now gene mutations can be categorized on the basis of the molecular nature, on the basis of the causative agent of that mutation, and also on the basis of what kind of phenotypic effect is shown. Always remember that there is a certain agent which will cause the mutation and in the genes, and this will be referred to as a mutagen. But normally also in the normal circumstances also without the presence of mutagen also the gene mutations can take place, and they do occur normally in every organism. So what kind of normal gene mutations take place? Let us try to discuss them first. See, there are two important types of base transitions. We already know that there are four important bases, that is A, G, T and C. A and G being the purines and T and C being the pyrimidines. Now what happens, generally A will double bond with T and G will have the triple bond with the C. Now sometimes in this particular sequence of the DNA where the nitrogenous bases are located, there might be some or what changes in these nitrogenous bases. Now suppose what happens, suppose here we have this purine. Now if this purine, let's say, is replaced by another purine, suppose this is our adenine and now guanine is replacing them, this particular purine. So this phenomena is called as transition. So in transition, one purine will be replaced by another purine, or one pyrimidine can be replaced by another pyrimidine. Now suppose this purine is replaced by another pyrimidine, that is, if this is adenine, it is replaced by either thymine or cytosine. So this condition is called as transversions. So always remember, transitions can also occur and transversions can also occur. But the frequency of transitions which are occurring is more than the transversions occurring. So in the nature, naturally transitions do occur, but transversions do not. Also throughout the lectures of mutations, I will talk about the permanent fixation of mutation. Now what is the meaning of that? See, I'll explain it in this part only, I'll clarify it. Suppose I have here this sequence and here I have this complementary sequence of this. Now suppose this A gene is replaced by G. Suppose if it is a kind of transition, what will happen? This will be replaced by another purine, and let me say the purine is G here. Now we know that this is not correctly incorporated, but as soon as the two strands separate, this strand will act as a parental strand and this strand also. Now suppose I say this strand is this. Now a new strand will be formed. That time it would be like this. Now you see, for this incorrectly incorporated nucleotide, the complementary nucleotide is formed. Now as soon as the new strand is formed, now this time there can be no changes because it is correct in its own way. And that time we say that the mutation is fixed permanently. Now after this the mutation cannot be changed. But until this fixation has not occurred, till that time it is temporary mutation, it can be changed. So this is what I will be talking about throughout my mutation lectures. Okay, moving on further. Now here I have to talk about the first of all was the base substitution that I have already talked. Second is insertions and deletions. Now insertions and deletions refer to where a group of nucleotides can insert themselves or delete themselves. Now this is a very common sense. If 1, 2, 4 or more than that, these nucleotides are inserted or deleted, they will cause the reading shift in the reading frame from the point where they are inserted. We know that the reading frame is always in the form of triplets, 3 nucleotides at one time we read, and even this is read by the ribosomes. So if one of them is inserted, then the whole of the reading frequency frame will be changed. So this is referred to as frame shift mutation. But in case if three nucleotides or in multiples of three, suppose 6 or 9, if this are added, they will only add a new codon. They will not shift the frame. Still the three nucleotides will be read together, only one extra codon with three nucleotides will be added. So that is not a frame shift mutation. So always remember that not all insertions and deletions lead to the frame shifts. And those mutations which do not affect the reading frame are called as in-frame insertions or deletions. And these nitrogenous bases which are added, maybe these kind of or maybe these kind of, they are collectively called as indels. So this may be asked to define that what are indels. So these are those nucleotide pairs which are inserted or deleted, which may cause the frame shift mutation or may cause the in-frame insertions or deletions. Moving on further, then we have the expanding nucleotide repeats as the third kind of natural mutation. In the expanding nucleotide repeats, what happens is that the number of repeated nucleotide sequences increases rapidly in the DNA. This was first observed in 1991 and this was observed in the X chromosome. X chromosome has a region which is known as FMR1 region. In the FMR1 region, there is a particular copy which is called as CGG. This is a codon. And when its multiple copies are present, ideally there should be some 50 to 70 copies of this present. But what happens during the mutation? This is present in hundreds to thousands of copies. And that is the condition when the chromosome looks very large in the size. So this causes the fragile X syndrome. And this is the expanding nucleotide repeats perfect example. Okay. Moving on further, we have to talk about the these all are actually not in the course, so I did not discuss this. Okay, there is somewhere the term given which is referred to as anticipation. So we have to also learn that what is the meaning of anticipation here. I'm just searching for that term, anticipation. Okay, let's discuss it ourselves. So anticipation is a phenomena where a particular disease becomes more severe in the successive generations. So suppose there is a parent here, this is a parental cell and this consists of a mutation. Now when this will be transmitted to the progeny will be also having the mutation. But as the more and more mutations are transited this further into the subsequent generations, the phenotypic effect which is shown by that mutation becomes more and more intense. And that is the condition where we say that this is your anticipation. Okay. Now we have to talk about the yes. See, mutations can result from both internal and external factors, those that occur under normal conditions. As I was talking about this also, the normal condition mutations which occur, they are the spontaneous mutations. Spontaneous means autonomously occurring. While those mutations which are induced by certain environmental chemicals, they are referred to as the induced mutations. So spontaneous mutations and the induced mutations. Now we have to discuss both of them one by one. Let's talk about first the spontaneous mutation. So see what happens in the spontaneous mutations, there is generally the change in the base pairs. The base pair, one base pair will change another one. Maybe transition can occur or maybe transversions can also occur. But yes, these will be occurring. Now the main reason is because of the tautomeric shift. Now tautomeric shift is, suppose there is particular, this is the particular base and here we have the hydrogen attached. But due to a shift, this proton is shifted here. Now with that condition, the two bases change completely, and because of this shift, the insertions and deletions also uh, have to happen accordingly. So that is what the tautomeric shift is called as. So tautomers are nothing but the different chemical forms of a nitrogenous base which differ only in one proton position. And due to the tautomerization, there can be the incorrect base pairing, and they will also then not follow the Watson-Crick model. Now always remember that tautomers are extremely rare and unstable because this proton cannot be residing here for a very long period of time. So the tautomeric shifts are also very rare because the tautomers so formed are very unstable. Let us take one example here. Hmm, See, suppose this we are taking as thymine. So thymine has this kind of structure. So here we have the nitrogen attached, here also we have the nitrogen attached, and thymine is nothing but 5-methyl uracil. So here we have O and here we have O. Now this is a normal structure of thymine. Now here the H which is added here is moved on to this O. So this forms this kind of structure. Now this is a keto form. This is a keto form of pyrimidine. It is converted into enol form. So always remember, in the tautomers, the keto form of the pyrimidine is converted into the enol form. And similarly, there is amino form of purines. It is converted into imino form. We will also have one of the examples here. Yes. So this is your amino form and this is your imino form. So this can also occur. So this is all because of the tautomers so formed, but these tautomers which are formed, they are very unstable in nature, and because they are unstable in nature, the tautomeric shifts are also very rare. But earlier they were proposed in a way that they do not follow the Watson and Crick model and all, but this is not the actual reason for the spontaneous mutations. Earlier the scientists have believed that tautomeric shifts were also the important phenomenon of the spontaneous mutations, but nowadays there are other reasons also, one of them being the wobble base pairing. So what is the meaning of wobble pairing? See, sometimes what happens is, we know that DNA structure is right-handed helix, and this DNA structure is very flexible in nature. Because of this, sometimes a correctly inserted nitrogenous base can also show some kind of errors, and instead of that nitrogenous base, another nitrogenous base can also be fixed. So that is also some kind of mistakes can occur because of this flexible nature of the DNA, and this is known as the wobble pairing. And they have been even directly detected in the DNA, and they are the natural major causes. What happens generally? The adenine here is replaced by guanine, means the purine is replaced by purine, or the thymine is replaced by cytosine, all because of the flexible nature of DNA. So this condition is known as wobble base pairing, and this is more natural and this is more frequently occurring rather than the base pair, or what do we say, tautomeric shifts. Then after that there can be the replication errors also which can occur. See, sometimes if I say that the vowel base pairing has occurred, so in vowel base pairing supposedly with the thymine, suppose let's say after this thymine the guanine has been inserted in the place of thymine. Now if this guanine has been inserted, what should happen? After this, see, this is thymine and this has to base pair with adenine. We know this, that thymine will base pair with adenine. But because of the vowel base pairing, instead of this adenine, guanine comes into the position. So because of that, now this parental strand here, whatever this parental strand will be serving as the parental strand, the new strand form will be completely looking like normal, and in this condition this will be called as a transition mutation, wherein this mutation will be permanently fixed. So I'll again explain it. What is happening? Suppose we have this kind of sequence, or let's take this sequence only. Suppose we have TTCG. Now what should be the complementary strand to it? It should be uh, It should be A, G and C. Now suppose during the because of the flexible nature of DNA, this A does not incorporate, and rather than this there is a G incorporated. Now we know that this G is incorrect, but supposedly this now separates and both of them are acting as a parental strand, TTCG. So here it will be forming AAGC, and here this is AGGC, which is incorrect. Now this will form TCCG, but this TCCG is very correct, because according to it, it is correct only, no? So that time this is what we call as the transition mutation, and we say that the mutation is fixed permanently. So this can also be one of the uh, spontaneous mutations which can be occurring. Now there can even be some of the spontaneous chemical changes which can be occurring within these strands. So this is also one of them, that there is some kind of spontaneous chemical changes. Now what happens here generally, suppose let's take the example here. Let's say there is a ribose sugar here, and this sugar is attached to adenine, this adenine being your purine. Now in the particular sugar phosphate backbone, suppose here is your sugar and here was your adenine, and here were the another nitrogenous bases. Now suppose due to one or the other condition this adenine is removed from its place. So now when this adenine is removed, this creates an apurinic site, and during the replication this apurinic site so formed will be randomly filled by the DNA polymerase. So now here we know that earlier adenine was present here, but now in the place of adenine any random, here earlier adenine was present but now adenine is removed, and instead of this adenine any random purine can be inserted by the DNA polymerase. Now maybe it be adenine, may it be thymine also, sorry, guanine also, anything can be inserted by the DNA polymerase. So this is a spontaneous chemical change which has occurred. Now suppose here the guanine was inserted. Now after that this will be serving as a parental strand, and when the transition mutation will occur, the mutation will be then fixed permanently. So this is all because of a spontaneous chemical change. So even this can occur. So in the next video we will talk about the non-spontaneous or the chemically induced mutations. We will talk about how the chemical mutagens and the physical mutagens can also bring about the mutations.